Okay, so welcome to this video. In this video what we're going to discuss is the transduction of painful stimuli. Okay, so this video is going to be on the transduction of painful stimuli, which means how we turn painful stimuli into action potentials which will fire within nociceptive neurons. Okay, so transduction of painful stimuli. So the structure then for this video is we're going to start off by looking at nociceptive neurons, okay, which are called nociceptors, and I'll just discuss in a moment the definition of nociceptor versus nociceptor. We will see the anatomy and the physiology of these nociceptive neurons, and then what we'll move on to is the proteins which are actually involved in triggering a receptor potential within these nociceptive neurons in response to the nociceptive stimulus. Okay, right. So let's begin then with the nociceptive neurons themselves. Okay, so we're going to start off with a little bit of terminology then. And then what we'll do is we'll move on to look at the anatomy of the skin, look at the anatomy of these nociceptive neurons within the skin. Okay, right, so we'll start off with what the difference between a nociceptor and a nocicensor is. Okay, so we're going to start off with this distinction. So, a nociceptor is the name for a neuron which detects painful stimuli. Okay, so this is a neuron which detects painful stimuli, what I've previously just been calling a nociceptive neuron. Whereas a nocicensor is a protein which detects well, not necessarily a protein, but some sort of receptor, basically, protein/receptor. They are going to be proteins, the ones we're going to look at, okay? So the protein/receptor which detects the noxious stimulus, okay? And that's another little piece of terminology we could define. A noxious stimulus just means a painful stimulus. Okay, so noxious is just a fancy word for painful. Right. Okay, so that's the difference between a nociceptor and a nocicensor, and that's an important distinction because people often get confused and think that nociceptor must mean the receptor because you've got the septor bit at the end. Okay, the nociceptor refers to the nociceptive neuron, the whole neuron which detects the painful stimulus, whereas the actual receptor which detects the painful stimulus that is within the neuron and therefore is going to transduce the noxious stimulus into an action potential within this nociceptor is called a nocicensor. Okay, right. The other little thing that I want to clear up, which we're not going to dwell on hugely in this video, but I just want to say it anyway, which is the difference between nociception and also pain. Okay, so nociception refers to the turning of a noxious stimulus into an action potential within a nociceptor. So this means the transduction of a noxious stimulus into an action potential, basically. So it just refers to something which is happening within this nociceptor. It refers to the mechanisms within the nociceptor by which a noxious stimulus is turned into an action potential firing within that nociceptor. Pain is something much more complicated. Pain is the conscious experience, basically. Pain is the conscious experience associated with nociception. Okay, so when you have a noxious stimulus present in a portion of your skin, let's say, it will undergo nociceptive transduction, okay, so it will be turned into action potentials. That's the nociception. And now the effect that these action potentials actually have on the brain to produce a conscious experience, okay, the conscious experience of pain is what they will produce generally, but of course in certain times the action potentials coming in from these nociceptors will not actually produce pain. So nociception and pain are related concepts. Nociception will often lead to pain, but it's not necessarily a fixed rule that nociception will cause pain, and it's also not fixed that you need nociception in order to get pain. Okay, so there is such a thing as neuropathic pain, which is where the brain is producing this conscious experience of pain without the actual nociception, so without the nociceptors becoming activated by the painful stimulus. Okay, right, so let's now discuss the cutaneous nociceptors. So we'll begin by discussing the anatomy of the skin so that we can look at these cutaneous nociceptors within the skin. Okay, right. So the skin is made up of many different layers, okay? So the outermost layer of the skin is the epidermis, okay? So epi means around, and then it's around the dermis, so that's why it's called epidermis. Now, what I'm drawing in here is a hair follicle, which is a little invagination into the skin in which a hair will sit. Okay, so this outer layer here, this is all epidermis. Now, it's not necessary for our purpose to go into the detailed discussion of the different layers of the epidermis. Okay, there are 5 different layers of the epidermis, but we won't discuss that. What we do need to know is that the cells that are within the epidermis, well, the main type of cell that's within the epidermis, okay, so let's draw this cell here, is called a keratinocyte, okay? And these will actually become important later. So keratinocytes can play a part in the transduction of noxious stimuli into chemical stimuli, which can then go and act on nociceptors. Okay, so here is a keratinocyte within the epidermis. Right, then let's draw the hair within our hair follicle here. So this is going to be a hair here sitting nicely within the hair follicle, and it's sort of got this sort of bottom shape like that. Okay, so I'll colour in this hair in orange. Right, okay, so at the moment the hair is actually quite erect. Normally, of course, the hair would be more sort of slanted downwards. This suggests that I've drawn the skin of someone who's quite cold. Okay, so you erect the hair muscles to trap a layer of air over the skin and reduce the movement of the air over the skin to try and keep the same air in contact with the skin rather than having a continuously fresh air source coming onto the skin and therefore removing heat from the skin. Okay, right, so related to this there is a muscle which is involved in erecting the hair. Okay, so this is the erector pili muscle here. Okay, so it's a little smooth muscle piece here. So this is erector pili. It's not just one smooth muscle cell, it's many smooth muscle cells. And it will be activated to contract, and it will take the hair from an angle like that to this angle like that. Okay, so it will erect these hairs. Okay, right. So each hair within a hair follicle will have an erector pili muscle associated with it. Another structure that you can often have associated with hair follicles are things called sebaceous glands. Okay, so this is supposed to represent a sebaceous gland. And these secrete sebum, which is a sort of oil, the skin's natural moisturizer basically, into the hair follicle, and this will lubricate the hair follicle. Okay, so this is the sebaceous gland. Okay, right, so I have the sebaceous gland in purple here. Right, okay, so the layer of the skin underneath the epidermis is then called the dermis. Okay, and this is a layer of connective tissue basically, so you have a lot of connective tissue within this next layer. Okay, and the reason therefore that the epidermis is called the epidermis is that it's around the dermis, so the epidermis is the most peripheral layer of the skin basically, and it sits around the dermis. Okay, that's what epi means. So here is the dermis. So this is a layer of connective tissue, so you have a huge amount of connective tissue all over the place basically. Now, let's go a bit deeper before we discuss any other structures that we're going to have within the dermis. Okay, so underneath the dermis, what you then have is a layer of subcutaneous tissue, which mainly consists of fat cells. Okay, so you're going to have adipocyte after adipocyte after adipocyte after adipocyte. Okay, so this is the subcutaneous fat. Now, this layer of subcutaneous fat, this isn't a constant layer, basically. Depending on where you are looking at this section of skin from, you will have differing thicknesses of the subcutaneous fat layer. So, for instance, if you are looking at a portion of skin in the abdominal region, the subcutaneous fat layer will be hugely increased, basically, compared to, for instance, if you look at a piece of skin taken from the ankles. Okay, so the subcutaneous fat layer is the layer which is most variable depending on where you're actually looking at this piece of skin from. Okay, now in the subcutaneous fat layer, you have large arterioles and venules running. So let's draw one of these. So here's an arteriole, okay, and here's a venule. Okay, and these large arterioles will be giving off branches which will then go upwards and peripherally. So they'll go into the dermis and up and supply the epidermis. Okay, so this is an arteriole. Okay, and then of course you'll have little venules bringing the blood back to this bigger venule down here. So this is a big venule. Okay, so let's draw the branches which go from these two large blood vessels up into the dermis and then onto the epidermis. Well, they don't actually go into the epidermis, they flock underneath the epidermis. Okay, so here we have an arteriole, a smaller arteriole, which is a branch of our bigger arteriole, and then we also have the venule bringing the blood back into the larger venule here. And between those two, you will have a huge number of capillaries, and basically there is a massive great capillary bed sitting just underneath the epidermis. Okay, so the epidermis itself doesn't have capillaries within it. It's avascular, okay? However, you have a huge capillary bed sitting right underneath the epidermis, and this is what supplies the keratinocytes within the epidermis with blood, basically, with nutrients, and takes away the waste products. Right, okay, so those are other structures that you have, therefore, in the dermis. You have these smaller branches of these large arterioles and venules that are within the subcutaneous tissue. So, I'll now colour in the subcutaneous fat layer in yellow. Okay, so this yellow colour represents the many, many adipocytes that you will have in this layer. Okay, now let's discuss another structure that you might have here, which is a sweat gland. Okay, so sweat glands are basically little ducts which empty onto the surface of the skin up here. Okay, and the tube goes right through the epidermis, right through the dermis, and then you have the actual production portion right down here, which produces the sweat. Okay, so a little coil in this tube right at the bottom, and this is the actual sweat gland here. So this is the sweat gland which produces the sweat, and then the sweat is moved up the sweat duct here, and then it will go and be released onto the surface of the epidermis, and then what will happen is the sweat will evaporate from the epidermis, and when it does that, water, when it evaporates, requires a lot of heat to have been put in there, basically. It will take a lot of the heat from the surface of the skin, so if you're too hot, basically, you sweat onto the skin to cool yourself down. Of course, we've drawn the hair cell erect here, so we must be in a cold environment, so our sweat glands will not be very active at the moment. Right, okay, so that's another structure that you have, and it's important to acknowledge that the sweat gland bit down here is actually in the subcutaneous tissue portion. Okay, and then right at the bottom, down here, underneath the subcutaneous fat, you have a very, very strong portion of connective tissue, okay, and this is known as the deep fascia, okay? And then underneath the deep fascia, you'll start to get muscles. So, for instance, if you're in the abdominal region, you'll get the external oblique muscle and things like that. Okay, right. So this is the deep fascia. So this is very, very tough, very dense connective tissue. Now, between the deep fascia, which is a layer of connective tissue, and between the dermis, which is a layer of connective tissue, we have sitting this squidgy, squodgy subcutaneous fat layer. Okay, that doesn't seem very rigid. Okay, so to try and strengthen the structure up, what you have is within the subcutaneous fat layer, you have numerous ligaments, okay, little pillars of connective tissue, if you like, that stretch between the dermis and the deep fascia and hold them together, okay, to give a bit more rigid support to this subcutaneous fat layer. Okay, so these little pillars of connective tissue are known as skin ligaments, okay, and they have a fancier name also. They can also be called a ligament retinaculum. Okay, so you could call the skin ligament a ligament retinaculum, or if you like, ligamentum retinaculum. Okay, so ligamentum retinaculum. Right, okay, so those are the skin ligaments, and they're just to support the subcutaneous fat layer by having a connection between these two more rigid layers, which is the deep fascia and the dermis. Okay, so that's the sort of side bit of anatomy. Let's now talk about what we're really interested in, which are these nociceptors, which are going to innervate the skin, so-called cutaneous nociceptors. So basically, what you will have is a nociceptor looks like so. They're often called free nerve endings because what they will have is it's basically an axon with loads of little branches coming off it like a tree. Okay? And all of these little endings will have nociceptors in, so the actual proteins or receptors that actually detect noxious stimuli. Okay, and they're just sort of waiting to sense noxious stimuli. And if a noxious stimulus comes, it will trigger a receptor potential, which we'll talk about later, which can then trigger an action potential. And the actual site of action potential generation isn't quite known, but we'll discuss more of that later. Okay, so unfortunately the arteriole label is kind of pointing at the moment to our nociceptor here. But this sort of tree-like structure here, this was meant to represent our nociceptor. And if you like, if you want, you could call it, even in more detail, you could call it a cutaneous nociceptor because it's within the skin. So cutaneous just means pertaining to the skin. So a cutaneous nociceptor. Right, okay, so what sort of stimuli actually contribute noxious stimuli? What sort of stimuli would actually stimulate a nociceptor like this? Okay, so let's give some examples of noxious stimuli. So, some good examples are extreme temperatures. Okay, so either too hot or too cold. Okay, so generally hotness becomes painful at around 40 to 45 degrees Celsius. Okay, many people quote it at 42, 43 degrees Celsius. That's when heat starts to become painful. Okay, so at that sort of temperature, you will be activating nociceptors within the nociceptor terminals, basically. Okay, then in addition, too cold contributes a noxious stimulus. Okay, this one's much more variable, however. It's not fixed what temperature someone will actually find painful, basically. Generally, it's around 15 degrees Celsius, but you get some people who will find 30 degrees Celsius painful. Okay, so what I mean by this is if you do a simple experiment where basically you take someone's arm, so let's say here we have someone's arm, okay, and you put a little metal block onto their arm that has been heated up or cooled down to a certain temperature. So this block here is called a thermode. Okay, so it's a block of metal that's at a certain temperature. And if you put that onto their skin and then ask them whether they're finding it painful, then basically, with regards to it going too hot, there's a set answer. People find it painful when it starts to get to around 43 degrees Celsius. Okay, so there's a nice set answer in humans for this. Whereas with regards to too cold, what you can do is you can vary the temperature hugely, okay, and you ask them continuously, are they finding it painful? And you find that there's a huge variation in what people will find painful. So most people can tolerate down to 15 degrees Celsius before actually finding it painful, but some people will find that it becomes painful at around 30 degrees Celsius. So it is hugely variable. Okay, but hot isn't. Hot, there is a set point at which we start finding the stimulus too painful. Okay, right. Those are two noxious stimuli. Another obvious example of a noxious stimulus is mechanical pressure. Okay, so if you apply intense mechanical pressure onto the skin, then this can activate nociceptors, basically. Okay, and also there are chemicals. So I don't necessarily mean chemicals that you can put onto the skin. I mean chemicals that can be released by cells which signal that you have had tissue damage. Okay, so when cells are damaged, okay, they can undergo a process called necrosis, where basically they break apart and they release all sorts of substances into the extracellular fluid that would never usually be in the extracellular fluid. This sort of archetypal example would be adenosine triphosphate, ATP. Okay, this is a substance that is usually banished from the extracellular fluid. So if you've got ATP in the extracellular fluid, it is a giveaway sign that cells have died, basically. Okay, and that can also activate nociceptors. Okay, in addition, cells, remember, have a very high concentration of potassium within their cytoplasm, whereas the extracellular fluid has a much lower concentration of potassium. So if you break open a lot of cells, you're going to get the potassium that's within the cells released, and that will raise the concentration of the extracellular potassium, okay? And that can also activate nociceptors to fire. In addition, not so much around cell damage now, so this is cell damage here. These are chemicals which signal cell damage, but more widespread problem potentially. Okay, if you've got too high proton concentration, i.e. the pH of your extracellular fluid is too low, then that can also activate nociceptors. So if the pH is too low, so remember, proton concentration goes up, that causes pH to go down because pH is the negative logarithm of proton concentration. Okay, so if you plot a graph of that, what you basically find is if you put proton concentration on the x-axis here, and then we have pH on the y-axis, what you get is a graph that looks like this. Okay, so as proton concentration goes up, pH goes down. Okay, and it can become negative if you take your proton concentration high enough. Not under physiological conditions, of course, but in principle. Okay, basically, too high proton concentration can also activate nociceptors. Right, so that's we'll call it there for this video, and in the next video what we'll discuss is the anatomy of these nociceptors, so the different diameter fibers that you can have, and also where their cell bodies are and how they enter the spinal cord.